This is Dr. Sriram from SNS College of Technology. In this session, we are going to discuss about atomic absorption spectroscopy. Introduction about AAS is a spectro-analytical procedure for the quantitative and qualitative analysis of chemical elements and chemical substances. Is based on absorption of light by free metallic ions that have been atomized from a sample. In this technique is used for determining the concentration of a particular element in the metal, heavy metals like copper, iron, zinc. So that kind of heavy elements we are analyzed from this technique. This shows the modern instrument of AAS. Principle of AAS. The technique makes use of a atomic absorption spectrum of a sample in order to assess the concentration of specific analytes within it. It requires the standard with the known analyte content to establish the relationship between the measured absorbance of the analyte concentration and relies therefore on Beer-Lambert's law. So Beer-Lambert's law is a very important law to understand the concept of AAS. The atomic absorption spectroscopy measures the concentration of specific elements in a sample by analyzing the unique fingerprint in the form of atomic absorption spectrum. So here the step 1, the sample preparation, and step 2 is the atomization. So this is the first step. The sample is typically dissolved in a suitable solvent. Most of the heavy metals are dissolved in the acids with help of the deionized water to create a liquid solution. This ensures the analytes are present as free atoms ready for absorption. So that is we go for always the liquid form of sample, use it for this technique. For solid samples like ores or minerals, additional steps like grinding and digestions with concentrated acids may be required to break down the matrix and liberate the analytes. So most of the sample preparation technique we can use it for the acids with help of the deionized water to digest the samples. Sample 2, step 2, atomization. The prepared solution is nebulized into a fine mist and introduced into a high temperature flame with help of the air acetylene as a fuel. This intense heat in the flame excites the electrons in the analyte atoms, promoting them to higher energy levels. Step 3, the absorption process. The simultaneously a hollow cathode lamp containing the same element as the analyte emits a specific wavelength of light. So we are use a source of hollow cathode lamp, almost range of different heavy metals. We can use it for the hollow cathode lamp that corresponds to energy difference between the excited and ground state of the analyte atoms. As the emitted light passes through the atomized sample, some photons are absorbed by the excited analyte atoms, causing them to return to their ground state. This absorption decreases the intensity of the light at the specific wavelength, followed by the step 4, measurement and analysis. The light intensity before and after passing through the sample is measured by a detector. So this is a simple representation of the atomic absorption spectroscopy. So there is a hollow, uh, that is a light source. Then that light source is allowed to the flame. So that flame is created from our sample with the help of the gaseous fuels, air and acetylene composition. So and followed by the monochromator and detector and amplifier. Then we can get it the results from the display. So this is the basic concepts of the basic instrumentation of the AAS. The different components are used already. We discussed the light source, chopper, and nebulizers. So here light source is commonly used for the hollow lamps. Is used for the halo cathode lamps is used as a source. The chopper mechanically choked to the differentiate between the light from the source and the emission from the sample cell. Chopper distinguishes between absorption and emission signals. It is situated between the light source and the atomizer. Then third step, nebulizers that coming from our sample burner systems. It produces a mist or aerosol of the test solution. It converts the test solution to gaseous atoms. It sucks in liquid samples at a controlled rate and produces the aerosol spray for use in the flame. It effectively combines aerosol, fuel, and oxygen. So that once we pass through the our solution by using the nebulizers, so then go for the flame. So the atomization is happened here, process of separating particles into individual molecules and breaking molecules down into atoms. This is done by heating the analyte in a flame or graphite furnace to high temperature. So this step is very essential part. We can analyze our sample, convert it to the flame. Then monochromator, prisms, gratings. A line isolation device is necessary to ensure that only light of a wavelength specific to the analyte of interest is measured. A monochromator is a line isolation device utilized for atomic absorption. And then followed by the detector. The monochromator selected light is directed onto a detector. This apparatus converts radiant energy into electrical energy. That means it is in this step we are converted into the electrical energy, giving an electrical signal to the radiant absorption by the atoms. Typically the photomultiplier tube is used as a detector. Its function is to convert the light signal into electrical signal proportional to the light intensity. Then amplifier, the electric current from the photomultiplier detector is fed to the amplifier, which amplifies the electric current several times. It clearly we can get it for the results by using this step. And then a display. The applications of atomic absorption spectroscopy. Here we can understand the quantitative analysis. So how much concentration is present in the particular elements, and metallurgical and inorganic analysis. So this kind of analysis very useful from the mining industry. That means mining industry is getting the different ores that ores contain how much amount of the particular element. Then pollution monitoring. It is very important steps. We can collect the samples from the various places, liquids or gaseous form of the samples. We can convert into the liquid. Then we go for the analysis, how much amount of the heavy elements, heavy metals present the various medium, and also the food and beverages, and medical pathology. We can analyze the calcium, magnesium, sodium and potassium levels in blood serum and plasma are determined by using atomic absorption spectroscopy. And biochemical analysis for material analysis in agriculture field, and selected determination by atomic absorption spectroscopy while used for magnesium calcium concentration, vanadium present in the lubricating oil, trace elements in polluted soil, tin in canned fruit juice, chromium in metal, and many more analytical methods for the heavy metals.